Hi, welcome to the Instructor's video manual for cloning a fluorescent gene. I'm James Pyer from Gen.otyp, and I'm going to be walking you through 6 instructor training videos to get you ready to give your students an introduction to biotechnology with cloning a fluorescent gene. CAFG is a lab kit that's split into 6 45-minute sessions. It walks students through the foundational processes in the world of biotechnology today. It starts with PCR, polymerase chain reaction, which amplifies DNA, starting from a small number of copies to a billion or more copies. It progresses in the second day to ligation, where you take the DNA that you've amplified in day 1 and you place it into a bacterial plasmid vector, pasting in the sequence. On the third day, students do transformation, taking their completed plasmid and inserting it into E. coli bacterial cells. On the fourth day, they'll take the bacterial cells, now with the DNA inside of them, and grow only the bacterial cells that have the plasmid that they've inserted. They'll end the workflow with bacteria that express GFP, green fluorescent protein, that makes the bacterial cells that they've genetically engineered glow a bright green color under UV light. CAFG comes in 2 versions, standard and premium, which will both be covered in these teacher instruction videos. CAFG Premium includes 2 additional modules, an introduction to restriction enzymes and an introduction to gel electrophoresis, 2 other critical technologies in teaching biology to students. Whichever version of CAFG you're using, remember that it's designed to be difficult for students. Biotechnology is hard, and it requires concentration and attention to detail. Students that don't work carefully and purposefully may not have perfect results every time. But the students that try hard, understand what they're doing, and pay close attention to the protocols that we've provided will get good results. Both cloning a fluorescent gene standard and premium cover all of the requirements for AP Biology Lab 6, Part A, which normally just includes transformation. But these 2 labs go way beyond that. In fact, CAFG Premium also includes all of the requirements for AP Biology Lab 6 Part B, meaning you can turn the 2 AP Biology Lab 6 labs into 1 single continuous lab. At the end of each of the 6 chapters in CAFG, there are a set of pre-lab questions. We strongly encourage you to require your students to do these pre-lab questions before they come into the lab. That way, they already know what's going on in the test tubes that they're mixing, and it's not just like pipetting a clear liquid into another clear liquid. By forcing them to read the chapter and understand what's going on that day, they end up being more careful and getting more out of their lab experience. If you have any problems or questions about cloning a fluorescent gene, we'd absolutely love to hear about it and answer your questions. Please contact us through our website, gen.otyp.es. There you can find forums and contact forms to get in touch with us and answer your question quickly. Without further ado, let's get into the 6 teacher training videos that will take you through cloning a fluorescent gene.